My name is Walter Chazin. I'm a faculty member in the departments of Biochemistry and Chemistry, and I am the Chancellor's Chair in Medicine. I also am the founding director of the Center for Structural Biology and currently am director of the Molecular Biophysics Training Program and the Chemical and Physical Biology PhD program. So I'm a structural biologist, and we have two major research areas. One of them is largely called genome maintenance, and the other one in innate immunity. But what we do as structural biologists is determine the three-dimensional structure of biological molecules like proteins, DNA, RNA, carbohydrates. Those are the classic biomolecules. And we really strive to understand how these molecules work. So, for example, in genome maintenance, we're trying to understand how the many proteins that are active at a replication fork work together to replicate DNA. So I'm best known in my field for really in the beginning. I contributed significantly to the development of protein NMR, so the application of NMR spectroscopy to study in depth proteins. And over the course of my career, I initially was known for my work on calcium-binding proteins that translate calcium signals inside the body. And more recently, perhaps most well-known is our work on the iron-sulfur clusters, a special kind of cofactor, and how they may be involved in utilizing DNA as a wire to transmit information, electronic information, over long ranges in biology. So my approach to running a lab is built around the idea that my trainees should become independent scientists. This to me is the most critical aspect that I try and develop. And in order to do that, I must serve as a kind of mirror to help students, graduate students and postdocs and even undergraduates understand what are their capabilities, what are their strengths and weaknesses, and how they can apply and overcome their weaknesses and utilize their strengths to make the best contribution they can to advancing science. So my philosophy and approach for running a lab is really built around the concept of a family. And I really see my research group as my scientific family. And when I organize the group, I make sure that all the members of the group feel the same sort of obligation that you would towards your brother or sister who are a member of your family. That is, we always look out for each other. We're always there to be able to help each other. My lab, the idea of different students or postdocs competing with each other is absolutely a no-no. My laboratory uses a variety of different tools and methods to accomplish our goals. Of course, we're structural biologists, and we utilize a variety of structural biology techniques ranging from X-ray crystallography, NMR spectroscopy, small angle scattering, and electron microscopy in its various forms today. But in addition to that, we really focus on solving problems, and even structural biology typically does not provide all the answers. One needs to complement those studies with biochemical studies, with biophysical analysis, and even our lab carries out some experiments in cells, functional analysis. So it's really a broad range of techniques that are used in the lab. We really focus on the problems. So what attracted me to my area of research actually stems from a very early interest in molecular structure as an undergraduate. I am trained as a chemist, and so I always studied small molecules. Even in my PhD, I worked on determining the structure of small organic molecules known as natural products. And when I was finishing my graduate studies, the first papers that were published on applying NMR spectroscopy to proteins was published. And I was just fascinated by this because here there were really, really large organic molecules, and I already enjoyed studying natural products. So the shift up to proteins was just a natural progression. And from there, I've spent my entire rest of my career becoming a biomedical researcher, because when I started out, I really was a chemist. And hopefully I've made that transition over time. Basic science is important to my area of research because, in fact, I work almost exclusively on large teams that span the spectrum from the clinic to basic science. And I represent perhaps one extreme end of that spectrum, but I think one that is essential in the way that our teams approach problems. Of course, the funding is an enabler. It's an essential enabler, but it's an enabler. And what really is the most critical part is the people who are involved. Nevertheless, it's important, and this is, as a principal investigator, arguably one of the most important things that we need to do, and that's to create that environment, to run that almost small business, which creates the environment for our students, postdocs, and staff to be able to perform the research that is so essential to biomedical research progress. So success stories that have come out of my lab really range also a large gamut. I've had a long career. I think first and foremost, I would say the biggest successes out of my lab, perhaps most labs, are the scientists that we train. And I've trained over 100 different graduate students and postdocs over the course of my career. They're scattered throughout the scientific enterprise, from high school teachers to professors who I dare say are better known than I am, and I'm even proud of that. My lab determined the very first structures of classes of calcium-binding proteins, and in fact was the first laboratory to directly determine the effect of binding calcium on a specific calcium-binding protein. We were able to make critical discoveries in understanding where are the critical DNA binding regions in nucleotide excision repair protein that is central to the response to damage to DNA, a protein called XPA, and a number of other things, including, as I mentioned previously, this iron-sulfur cluster and its presence initially, and then generating some ideas about how it modulates the DNA polymerase alpha primase, a primary replication protein. So what attracted me to Vanderbilt ultimately was what we call the secret sauce, and that's the collaborative nature that is intrinsic to the way this university operates. I was actually recruited here to start a program in structural biology. And in the vision that I developed at that time, it was 1999, actually, I thought about what the future in structural biology would be like. And I felt it was important that we generate this new program around a forward-looking, almost avant-garde theme of integrating the techniques of structural biology to address basic problems in medical and biological research. In order to do that, we needed to create a different kind of environment, one that mimicked what was already here, this idea that research groups readily collaborate. And when I came and visited here and I saw how open the labs were and how effective the collaboration was, I kind of knew that this was really an ideal place to develop this theme, to develop around this theme. And so ultimately it was the combination of the offer, of course, the idea of coming here to create a new program, and the environment or the intrinsic environment that there was about collaboration. What distinguishes Vanderbilt from other institutions, besides its culture of collaboration, is the close geographical localization of engineering, science, medicine, and biology. We're all just 5 minutes away from each other, and that's an amazing juxtaposition of these different areas of expertise, and which is really effective at promoting team-based science, which is clearly the direction of where the most impactful research is today in the biomedical research arena. There's no question about that. So my advice for students and postdocs entering the field is to find your passion. Find something in science, in this area, that you enjoy, that fascinates you, because this really can become a lifetime pursuit. And as a career, working in science is a great treasure, because since we spend so much of our time during the day working, no matter what our career is, if you have something that you really enjoy to do and that fascinates you, you'll always be happy, or at least for the most part, be happy. Of course, there are other life challenges, but this really puts a positive spin on your life. And for me, I love what I'm doing. And even after all these years, I still am anxious to come up the stairs. I don't walk slowly, trudge up the stairs, but I'm jumping up the stairs as much as I can, and can't wait to get into my office, into the lab, and meet with people and start thinking about the next challenges that there are confronting us. And this kind of intellectual stimulation is really a treasure that carries a career just a very long way. So when I recruit a student or postdoc, one thing I look for is some degree of enthusiasm for what we're doing. And another aspect I think that's important in a scientist is that they be organized. And organized in their approach, organized in the way that they might make a presentation, organized in the way that even they keep the area around their bench. These are positive attributes that help a scientist be successful. And so that, and in the case of a postdoc, I also look to see their track record. And it's a little bit harder to evaluate in an incoming graduate student, but in a postdoc, they've done a PhD. I look very carefully at their track record of productivity and to find evidence that they not only carried out some research, but they were able to complete that research and publish that research. So diversity in the research environment is absolutely essential in my view. And throughout my career, having been in a number of institutions, diversity means a lot of different things. Part of it is just in the way that we approach our science. As I mentioned earlier on, our laboratory doesn't utilize one technique to look at one specific question. Rather, we use a diverse set of techniques and approaches to address whatever problems that we're trying to solve. And that's just at the research level. Another aspect of diversity, which to me has always been important, is to run a laboratory that had diverse perspectives. So this is not just having structural biologists join my lab, but students and postdocs coming from a chemistry background, from a biology background. Actually, one of the greatest things that I've seen over the years, one of the biggest helps to my lab is when we have a biologist among our midst. Right now we have a cancer biologist who's working in our lab, which has a lot of chemists, otherwise chemists and biochemists and structural people working in the lab. But the impact of the biologist, the different perspective of the biologist is very, very important. A third component of diversity, which is, I feel, super important, is the diversity within the people who are working in the lab. I've had the pleasure and the privilege to have students and postdocs coming from all corners of the world working in my lab. And this has always been very important to me, that people bring different cultures and different languages to the lab so that we are exposed and get to understand and to benefit from the different perspectives that people have. And I think the last component to the diversity that I'll stress is that even among the Americans, it's important that we have diversity within our laboratories. And I've always strived over the years to generate a ethnically diverse, a racially diverse, and as much diversity as I can through various connections and collaborations and links that I've built up over the years. And I think in this sense, I am someone who really treasures diversity. In these senses, in these 4 different ways, I really do treasure diversity. My academic and training background was not a straight line, and I think that this really benefited me in the end. In fact, I went to 4 different universities for my 2 degrees, bachelor's degree and PhD. I wound up in Canada and created a lifelong bond. Found my wife, who was my lab partner as an undergrad. And then I started a PhD program. And after 2 semesters, I left. And it took me a few years to get back to graduate school. But ultimately, I then went through graduate school very quickly. And all of this was a great benefit to me. So I went to Carnegie Mellon, then McGill University, then University of British Columbia. I finished up my PhD at Concordia University, the last 3 all in Canada. Then I went to Zurich for a postdoc and worked for the person who was involved in developing protein NMR, who ultimately won the Nobel Prize, Kurt Lüttich. And from there, I took a second postdoc at Scripps and was promoted. After about a year there, I was promoted to a faculty position and held that faculty position for 12 years until I was recruited here. Because I look upon graduate students and postdocs and look to see whether they have the benefits of those difficult roads. Because in fact, that focuses you, that hardens you. And if we were to put it in a semi-comical way, I'm the sucker who always falls for the black sheep because I myself was one. And I don't give up on people just because they had a bad experience. And in fact, I had the only MD, PhD student who ever took a sabbatical during the middle of his MD, PhD training. He took a year off. But in fact, he proved to be very successful. He graduated, he is now on faculty, and he's an associate professor there. So, and I guess my story kind of parallels that in some ways, and here I am. So I trained all these people over the years, and there's so many examples of students who, of course, they're often very young. And the ones that I have had the greatest problem with are the ones who are most appreciative to me now because they didn't get what I was saying then. And, this guy who's very well-known professor in England, University College London, he always is telling me, I was so happy after I left the lab, I realized what you were trying to tell me. And now we're the best of friends. It's amazing.